Hey everyone, thanks for stopping by. Today I'm going to discuss some malaria data coming out of the Latin lab. So avian malaria is a really common parasite that's found in many different bird species all across the world, and some individuals are better than others at resisting infection. One potential way to reduce likelihood of infection is through investment in cellular immunity. But this does come at a cost because it's very energetically expensive and could potentially result in trade-offs with body condition. Currently, we have limited understanding of resistance mechanisms to malaria, so we decided to tackle that with an experimental approach. So our main question is, why are some individuals able to resist malaria infection better than others? And we predicted that individuals with high cellular immunity prior to inoculation are going to be more resistant to malaria infection, but they're also going to have reduced body mass and fat. So to look at this, we brought in wild sparrows and inoculated them with some naturally sourced plasmodium. We looked at several different immune measures and body condition. Here on the bottom left is an example of our phagocytosis and hemolysis hemagglutination assays. Up here for our inoculation procedure, birds on the left were inoculated with blood-containing plasmodium. And of those 14 individuals that were inoculated, 8 were successfully infected and 6 were able to resist infection. Over here on the right was our sham control group, and these individuals were injected with blood that did not contain plasmodium. So over here on the right, we were looking at mass of each group, each treatment group. And this was 13 days post-inoculation, when we would expect the parasite load to be at its highest. And we found that resistant and infected individuals had reduced body mass when compared to their sham counterparts. On the bottom, these are the data for our phagocytosis assays of both E. coli and S. aureus, and we found that there was no difference between these individuals and their phagocytosis scores. Top right, we're looking at pre and post inoculation for each parameter of immunity that we looked at, and we found no difference between any group. Below that, we looked at body condition and immune parameters, and we found, once again, no correlation between these. So in total, we found no evidence that immunity pre and post inoculation plays a role in likelihood of infection. And we suspect this might be because prior exposure to the pathogen might affect resistance. And we also found that fat was consistent between all groups, but body mass decreased between both these resistant and infected individuals, suggesting that the cost of resistance and tolerance might be similar. And finally, we found no evidence of a trade-off between cellular immunity and body condition.